Hello, everyone, and thank you for joining us for the third session of Top Crop Managers webinar series entitled What We Know About the New Midge Species. My name is Janin Belbeck, and I'm one of the associate editors for Top Crop Manager. I'm joined today by Dr. Boyd Mori, who is an insect ecologist for Agriculture and Agri-Food Canada. Boyd is a strong proponent of integrated pest management and has a robust background in insect behavior, physiology, and population genetics, which he uses to develop monitoring and management tools for insect pests. Boyd's projects revolve around basic and applied research questions using a variety of techniques, from field experiments to electrophysiological assays and next-generation DNA-RNA sequencing. He has active projects developing pheromone monitoring tools for both canola and quinoa pests. Additionally, Boyd is researching host plant resistance in both the swede and wheat midge, where he is part of a collaborative team whose aim it is to identify host plant resistance mechanisms against these potentially catastrophic pests. Before we get started, I'd like to remind everyone that this webinar is being recorded and will be made available to all attendees and registrants approximately 24 hours after this live broadcast. This webinar has also been approved for 1 CCA CEU in Integrated Pest Management. Further instruction for submission will follow the presentation. Just some housekeeping here. The session is scheduled to run for approximately 50 to 60 minutes. Boyd will speak first, and then at which point we will start the Q&A period. If you have any questions throughout the presentation, please type them into the questions tab found on the right-hand side of your screen. Just a reminder, this is the third session of our 6-part webinar series. You can sign up for the additional sessions at topcropmanager.com/webinars. So without further ado, I'll let Boyd take it away.
All right. Thank you very much, Shannon. And thank you all for joining me today. And taking time out of your day to listen to what we know about these midge species here on the prairies. So I thought we would start from the beginning and talk about what is a gull midge. So gull midge are these small gull-making flies. They're in a family called the Cecidomyidae. There's over 6,000 described species. So it's a very large and diverse group of insects. And they're all these tiny, minute little flies. Many are important agricultural and forestry pests. So here's just a few examples. These are specifically in the Contorania genus. So we have things like the Douglas fir needle midge, the sorghum midge, the pea midge, and one of the ones that I'll be talking about today is the swede midge. Now, I'm sure most of you are familiar with a few different midge species. And midge is kind of a generic term. There's also another group of midges which are biting midges, but we're going to avoid those today and talk about these gull midges. So there's a few important gull midges in Canada, and the primary ones of importance for field crops are these three here. So I'm sure most of you are familiar with the wheat midge, a common pest on wheat. There's also the Hessian fly, which can be a sporadic pest on wheat and other grasses, and then swede midge, again, which will be the focus of today, and that's a pest of crucifer vegetable crops as well as canola. So we're talking about gull midges, but what are gulls? So gulls, they can take many different shapes and sizes. So these are really abnormal swelling or protrusions of plant tissue that's caused or can be caused by a variety of different parasites, including things like fungus, bacteria, lots of different mite species, as well as insects. And so oftentimes it's difficult to tell what made that gull unless you can dissect it open and actually see, you know, is there a little insect inside or is there a mite in order to identify what caused that gull. In insects, often the gull is initiated by specific proteins or enzymes within the feeding insect saliva. And so these can trigger a response in the plant and can often co-opt the plant mechanisms, growth mechanisms, and force the plants to actually make these gulls. So these gulls offer a nutritious food source for the developing insect, as well as they can provide protection of the insect. Now on your screen, these are a few different examples of gulls and what they look like when they're open. But because these are insects and they're not all created equal, we have some gull-making insects that don't actually make a true gull. So the plant tissue doesn't fully enclose them, and it might just become, swollen or distorted, maybe a little bit twisted. And if you're familiar with damage caused by the wheat midge or the Hessian fly, those don't really cause a true gull. But their feeding characteristics, in which they release this enzyme onto the plant, is very similar to other gull-making species, and that's what can trigger the shriveling of the kernel in wheat midge and the stunting of the plant with the Hessian fly. So we're going to focus on a couple other midge species. So swede midge is going to be the one we're going to start with today. So this picture looks quite large on your screen, but in reality, this fly is only about 2 to 3 millimeters in size. So it is very tiny. It has many different host plants, all within the Brassicaceae family. So this includes things like broccoli, cabbage, cauliflower, Brussels sprouts, bok choy, all those different crucifer vegetable crops, as well as canola, mustard, and other brassicas, including weeds. So, stinkweed, peppergrass. Those are all potential hosts for the swede midge. So it's native to Eurasia. We're not exactly sure of its exact origin, but it's not from North America. It is invasive. It was first introduced into North America, first recorded in North America in 2001 from Ontario. So a lot of insects are usually introduced into the US, but this is one we actually got first. Now, I should say it's recorded, its first record is from 2001, but damage symptoms were seen as early as 1996 in Ontario on crucifer vegetable crops. So it has been present in Ontario for quite a long time, but it wasn't until 2001 when it was first positively identified. On the prairies, the Canadian Food Inspection Agency first identified swede midge in 2007 in Saskatchewan and in Manitoba. So it has been identified from the prairies. Now, once it was identified in Ontario, there's active research being undertaken there by Dr. Rebecca Hallett at the University of Guelph, and as well as some researchers with Agriculture and Agri-Food Canada. One of the first things that they looked at was the potential range and where swede midge could exist on the prairie, or throughout Canada. So they conducted a study. They looked at bioclimatic modeling. So this is using things like temperature, moisture, and habitat suitability to see where swede midge might potentially be able to expand into and occur. So this map just indicates where our favorable conditions exist throughout Canada. And so what you can see, the green, those are unfavorable conditions, whereas then we get into the yellow and the orange, they become suitable, favorable, and then very favorable in the red. And so what you should see right away is that Southern Ontario, these are very favorable, this is a very favorable area for swede midge. And this is the region where it was first identified. So there's lots of crucifer vegetable production in this region. But what you should also notice is that a wide swath of Canada is a potential, or there are potential suitable climatic regions that swede midge can occur in. And I'm sure you're all familiar with the major canola growing region on the prairies. It's all considered suitable based on these models for swede midge. So this was a good piece of evidence for further support that we need to study swede midge and how its range might expand in Canada. So here's just a map of the invasion of swede midge in North America. So by 2000, we knew it was in Ontario. 2001, it was also identified in Quebec. From there, it spread into New York. And then 2005, 2006, it was found throughout several more eastern seaboard states, including Vermont, Massachusetts, Connecticut, and New Jersey. Then it was found throughout more provinces in Canada. It was identified in Prince Edward Island, Nova Scotia, Saskatchewan, and Manitoba. And it was at this time that the USDA actually approached the Canadian Food Inspection Agency about deregulating the status of swede midge. So before this time, it was considered a quarantine pest, but then in 2009, it was deregulated and is no longer considered a quarantine pest in both the US and Canada. Since that time, it's spread a little bit further into Ohio in the US. In 2015, it was positively identified in Michigan, and in 2016 in New Hampshire and Minnesota. So you can see it's making kind of an eastward and westward progression throughout the United States. In Minnesota, it was confirmed in vegetable plots right in Minneapolis. So it is making that westward expansion. And this is presently where it stands. This is the known distribution of swede midge throughout North America. Now we can think of, all right, well, what's the potential impact on canola? So here's the primary spring canola production region in North America. And if we overlap that with our our known distribution of swede midge, you can see that there's quite a bit of overlap. So there is a huge potential for swede midge to affect our canola crops. So let's talk a little bit more about swede midge biology and what makes it so damaging. So swede midge, swede midge, they overwinter in the soil. Okay, so they're in these cocoons here up the top left of this picture. So they're covered in soil. They look just like little tiny clumps of dirt. Inside, they will pupate. So that's what these yellow things are here on the bottom of the picture. Those are the pupae. They'll pupate in the spring. The adults will emerge. The adults are very short-lived, so they only live for about 1 to 4 days in the field. They mate. The females lay eggs on the actively growing portions of the plant. So if we're talking canola, this can be the primary meristematic tissue, but it can also be on secondary or tertiary branches or racemes of the plant. Those eggs hatch in about 1 to 10 days, and then the larvae develop. And so the larvae will feed for about 7 to 21 days before they will drop from the plant, move to the soil, and pupate. So part of the reason that swede midge can be so damaging is that in Ontario, there can be 4 or 5 overlapping generations depending on the temperature and moisture. So all these overlapping generations really allows the populations to increase quite readily and can really affect the plant. And so specifically on canola, what can it do? Well, larval feeding can really distort plant tissue. So here's just some examples of what it can do. The top left here, you can see this really severely damaged plant. So you can see the pods and the flowers, they're all growing from that single point. And that's where the larvae are going to be feeding in. They cause a lot of tissue distortion and especially the primary bud cluster up on the top right here. You can often see what we call this palm tree effect, in which the meristematic tissue on that primary raceme has been killed, And so the raceme no longer elongates, and all the pods kind of come off at the same portion of the stem, making this palm tree effect. And then we also can have just crumpled leaf tissue shown here in the bottom right. And so swede midge damage really depends on the growth stage of the plant. And the earlier swede midge attacks the plant, the higher the damage can be. So if swede midge really attacks in kind of the 3 to 7 leaf stage, it could wipe out the primary raceme, and then you have to hope that there'll be secondary and tertiary racemes that will help offset that yield loss. But here's a picture of a field in Ontario in 2013 in New Lescard. So this is the northeastern region of Ontario, and this is a small canola production region, but this region was really hit hard by swede midge. And so by the end of July here, this full field should be flowering, but you can see this large section is not close to flowering. This was caused by swede midge early and in high numbers, which basically took out all those primary racemes and even some of those secondary growing points. And this field basically had very little yield off of it. And now swede midge populations in this region, this New Lescard region, actually became so high that the local canola growing group actually asked for a 3-year moratorium on canola production, voluntary moratorium, to try and reduce the level of swede midge populations. Canola production has resumed in this region, and it does seem to they can produce canola with decent yields now. So what makes it swede midge so damaging? As I pointed out, swede midge can attack many different growing stages of canola, and because of these multiple overlapping generations, canola is vulnerable for a really long time period. So late vegetative, so anywhere from like the 3 to 7 leaf stage, canola can be is susceptible. The early bud stage, that's probably one of the most damaging stages. If swede midge females lay their eggs on the early bud, this can wipe out the yield from this plant, as well as all these secondary and tertiary buds. Those are also very suitable for egg laying and larval growth. So all of these points can be attacked by swede midge, and that's what makes canola so vulnerable. So, depending on plant stand density, sometimes, a lot of your canola yield will come from in a very dense stand, most of your canola yield will come from that main raceme. But if you have a less dense stand, you will get more yield or compensating yield in those secondary or tertiary racemes. And so all of these can be impacted by swede midge, and they can all cause significant yield loss. So what can we do about it? The picture doesn't look that good, but there are some things we can do about it. So swede midge, we can monitor for it. There's a pheromone monitoring trap available. So they're highly effective at capturing male swede midge. So right now, the recommended protocol asks for 4 traps per field, spaced approximately 25 meters apart, at about canopy height. And there is a nominal threshold. And so basically, after there's 1 true leaf on the plant, you're supposed to count the number of midges until you've captured 20 total midge across all the traps. And that's when you would do your first insecticide application. And then after that, the threshold changes a bit. Then you look at the number of midge captured per day per trap. And if you're averaging 5 males per day per trap, then you might have to do another insecticide spray. So there are 2 registered products that are for swede midge and canola. So both Matador and Coragen are registered. And for more information, I'd really refer you to this bottom link here, the swede midge and canola info sheet. there's a lot of good information there. But it is really dependent on growth stage, and once canola is past, once those buds are formed on those secondary and tertiary branches, we think canola is really no longer susceptible to swede midge damage. So what else can you do? Well, crop rotation is really key. So a 3 or 4 year rotation is highly recommended. So the reason the 3-year moratorium on canola production was asked for in that northeastern Ontario region is that it's found that swede midge can actually overwinter in the soil for longer than 1 season. So if we only went to if they only asked for a 1-year moratorium, there still might be swede midge present in the soil. So a swede midge can be in the soil for probably 2 or 3 years. So we really need a longer crop rotation to help reduce these populations. It's also, if you can avoid planting canola near a previous year's canola field, that also helps reduce populations. So swede midge, they are very small, they are weak flyers, but they could easily, cross a road or fly a few hundred meters. So again, if we could space out our fields, I know this is not always possible, but the greater the space between the fields, often the better. Removal of volunteer canola in ditches, including other brassica weeds that could act as host plants. This can also help reduce swede midge populations. Tillage may help reduce overwintering populations, but that, of course, is going to be up to the individual farmer and if they want to till or not. Overall, it might not be the greatest control method. Early plantings appear to be less damaged. So if we can get the plants through kind of the highly susceptible early growing stages earlier in the season before there's large swede midge populations, that lends itself to increased yield. And of course, there's 2 insecticides that I mentioned. And of course, we want to use our due diligence with those. Preferably use the pheromone monitoring system to help time those applications. And we want to be rotating our active ingredients for those products Okay, so that's kind of the state of swede midge and how it's what it's like in Ontario. But when CFIA found or discovered swede midge on the prairies, that led some researchers here at Agriculture and Agri-Food Canada, Dr. Julie Soroka, to start studying swede midge on the prairies. And from about 2008 till 2012, she never found anything really of value. But in 2012, there was thought to be some swede midge damage in some producer fields in northeastern Saskatchewan. And that's when a major research program was implemented. And so this research in Saskatchewan was led by Dr. Julie Soroka and Dr. Lars Andreasen, her postdoc. And they were surveying for the distribution of swede midge. So they were using swede midge traps across, they sent them out to producer cooperators. Probably many of you on the line may have helped with this survey. They also were looking at its life cycle. So they used emergence cages to time and see when the midge were emerging from the soil in the spring. And they're also looking at potential biological control agents, if there were any in Saskatchewan that might be helping control midge populations. But based on this initial work, what they noticed were some significant differences from swede midge in Ontario and what they believed to be swede midge in Saskatchewan. And so what they noticed were, in Ontario, swede midge can have 4 to 5 overlapping generations. In Saskatchewan, they were really seeing 1 or 2 at most. Now, this might just be due to different climatic effects, but it could also be due to, the midge here. So if maybe it's a different biotype or a different race of swede midge that we have in Saskatchewan versus Ontario. They also noticed a few different physical attributes. So the ones in Ontario, they thought were a little more frail looking. Surprisingly, they had fewer hairs than those in Saskatchewan, which they considered more robust or larger and also hairier. The main damage symptom in Ontario was often these swollen and twisted primary bud clusters, whereas in Saskatchewan, really all what they were seeing were flower gulls. They could never positively associate any twisted or swollen tissue with swede midge. And in Ontario, they were really seeing really medium to extreme damage severity, whereas in Saskatchewan, we were really seeing none to very, what we would consider very low. So I was a recent hire in Saskatchewan in 2016. I was brought on to, part of my projects was to help finish up some work on swede midge. And we started to question whether or not the swede midge, or what we believe to be swede midge in Saskatchewan, was actually the same species as that in Ontario. And how this came about, so not only were there all these prior reasons based off of the number of generations, its physical attributes, its damage symptoms, and its severity, but we were also starting to think about its life cycle a little bit more. And part of the work we were doing, we had these emergence cages on 4 farmers' fields in northeastern Saskatchewan. We had 4 cages per field, and we would put these cages on canola stubble, so fields from the previous year in the springtime. And in 2 of these cages, we had a pheromone trap. That's what you see in the cage on the left-hand side. And then 2 of the cages on the field just were left empty. So how these emergence cages work is that the insect will emerge from the soil. They're positively phototaxic. They're attracted to light. So they fly up to where this white bottle is, up at the top. And in the bottle, there's a killing agent. There's a preservative, which will kill them and preserve them. And so in these traps, what we were seeing in 2016 was that what we thought were swede midge, the populations appeared to be increasing. So we were catching hundreds of males and females in these bottles at the top of these cages. But when we looked at the pheromone trap, so the sticky card that was in the pheromone trap, there was nothing on them. There's, and you think even by accident, these midges flying around the cage should fly into this sticky trap and be caught on it. But really, we were really seeing almost nothing on these pheromone traps. So my previous research was mainly on pheromones, and pheromones I know they're highly species specific, and they're highly attractive as long as you have the right pheromone. So they can attract species from hundreds of meters to their pheromone. And because this is what a female uses to attract a male mate, right? So if you have a large area that you need to attract a mate from, the female releases this pheromone. It's very species specific. It floats around the air. The male can sense it from far away, follow the trail, and find the female. And this is just a small enclosed area in this trap. So we would expect that if the males were really attracted to the pheromone, they would be captured here. But they weren't. So I was really worried. I was wondering if there was an issue with the pheromone. So, there could have been something wrong with the synthesis of the pheromone, and maybe it didn't work for swede midge. So I sent some of our lures to Ontario, to Dr. Rebecca Hallett, the University of Guelph, to test for us. Her and her staff, they put them out in the field, and within a matter of days, they caught hundreds of swede midge. So there was not an issue with the pheromone lures. So this had us really suspect. We're like, okay, is this really swede midge? So I also collected larvae to rear them out to adults. And so this plant on the left here, this is showing the main damage symptom we see here on the prairies. And that's what we call these galled or bottled flowers. Okay, so these flowers, they remain closed, they do not open. And if you open it up, you'll see these little larvae that's shown here on the right-hand side. So they're quite small, and these ones are pretty early instar. As the larvae get a little bit bigger, they get yellow in color. And if you've ever seen these in the field, you might be holding them in your hand, and one minute they're there, the next minute they're gone. And it's because these larvae can actually jump. And so that's how they kind of get out of the plant, they wiggle out of the plant, and then they jump off or fall off and move to the surface to pupate. So if you've seen them in the field and you think, oh, it was there and then it's gone, it's probably because it jumped off your hand. So we collected a bunch of these flower gulls and we brought them into the lab. We put them on top of some soil to let the larvae crawl into and pupate in, and then we looked at the adults that emerged. And so here's a representative female that emerged. This female is preserved in alcohol. So when they first emerge, it doesn't show up that well on this photo, but their wings are covered in quite a few hairs, and the pattern is no longer recognizable in this photo, but there is kind of a modeled pattern across their wings. And so when I saw that, I knew that wasn't a swede midge characteristic. So I took a really terrible cell phone photo down a microscope, and I sent that to Rebecca and Jamie at the University of Guelph, and I said, does this look like swede midge to you? And they both responded and said, no, we've never seen swede midge that looked like that. So they recommended that I send them some samples. And at the University of Guelph took a look, and he noticed some different characteristics between the swede midge in Ontario and what we thought were swede midge here in Saskatchewan. So one of these characteristics is, so on the left-hand side, we have a swede midge female, and on the right-hand side, we have a midge from Saskatchewan that emerged into our emergence cages. And so what we look at, if we're looking at the first two antennal segments on the left-hand side of the swede midge female, the first two antennal segments are about the same length as the third, okay? But if we look at our Saskatchewan female, if we look at those first two segments, they're only about half the length of the third, okay? And so these are kind of the little details that you need to be able to determine in order to tell if it's the same species or not. And so this was just one of the details that Jamie found that showed differences between swede midge in Ontario and this midge in Saskatchewan. Also looking at the males, he also noticed a few differences. So at the end of the male, at the end of the abdomen, there's claspers, which the male uses to grasp on and hold the female during mating. And he actually noticed that the shape of these claspers, they're slightly different in the swede midge males from Ontario and those from the males that emerged here in Saskatchewan. So both Rebecca and Jamie recommended that I send some samples to Brad Sinclair, who works at the Canadian Food Inspection Agency in Ottawa, also with the Canadian National Collection of Insects. And Brad is a dipteran expert, so he works on flies. And he followed up and took a look at these, and he noticed the same differences that Jamie did, and plus a few others. So he could confirm, in fact, that they were not swede midge that we were finding here in our emergence cages in Saskatchewan. And while Brad and Jamie were looking at the physical characteristics of the swede midge, I also was looking at a portion of its DNA. So I sequenced this 439 base pair region of what's the mitochondrial DNA, a gene, the CO1 gene, which is cytochrome oxidase 1. This region, if you've ever heard of DNA barcoding, that's what this region is. So it is thought that this barcode region, you can use it just like the barcode, on your groceries. You can figure out this sequence, this barcode, and you can compare it between different species, and it will tell you, as long as there have been previously identified species, what species it is. And this works better with some species, like animals, than it does in others. It doesn't work very well in plants. So when people say DNA barcoding, this is what they're talking about. And so you're comparing this barcoding region, so these letters, these DNA base pairs between different species. And so what I did is I sequenced some swede midge from Ontario, and I also sequenced the midge that emerged here in Saskatchewan. And you probably guessed it, they're different. So the top four sequences here, shown here, are C. nasturtii, which is the Latin name for swede midge, and then the bottom ones are our undescribed species from here in Saskatchewan. And so what you can see, anytime there's just a dot, that means it was the same base pair, same sequence. but if there's a change in letter, it shows up here, and you can see that all the Saskatchewan midges have several different changes in their sequence compared to Swede midge. And in fact, if you look at the sequence and compare all that whole length, there's only about 91% sequence similarity between Swede midge and the midge that we found here in Saskatchewan. And if you're really into DNA barcoding, depending on who you ask, they say anywhere you need about 98 to 100% sequence similarity to be considered the same species. So these would not be considered the same species based off of DNA barcoding. So all these lines of evidence, so we have morphological evidence, we have the DNA evidence, we have some behavioral evidence that this is in fact a different species. It's not Swede midge. So Brad in Ottawa was able to confirm that in fact it's in the Contarinea genus, so this is the same genus that Swede midge belongs to, but it is an undescribed species. So it has not been described from anywhere else in the world. Doesn't mean it's not found anywhere else in the world. It's just so far to date, it hasn't been described anywhere else. So you guys were probably all hoping to get a new name for this species. Right now, I'm going to call it to be determined, except I didn't write that acronym right. But anyways, so the name will be published hopefully shortly. Brad is working on finishing up his species description. And once that is out, we will submit it for publication and we'll have a formal name. And for the rest of the talk, I'm just going to refer to it as the canola flower midge, just for ease of reference. So we know it's a new species. We know so far it hasn't been described anywhere else. And we had the several years of research that Julie and Lars had conducted on what they thought was Swede midge here in Saskatchewan. And when we went back and looked at their samples, when I looked at their samples, I actually found that they weren't, the ones that they were collecting in their emergence traps were this canola flower midge. They weren't Swede midge. And the ones on the pheromone traps that Lars had and he believed were Swede midge, in fact, most of those were not Swede midge. So if you Google Swede midge right now on the prairies, you might come across several maps with pheromone, where pheromone traps were placed out and they indicated that they were positive for Swede midge. I would say that 99% of those are not actually positive for Swede midge. And so personally, I would say that we haven't had a positive identification of Swede midge in a pheromone trap since those initial Canadian Food Inspection Agency traps in 2007 and 2008. But looking at Julie and Lars' data, plus some data that we've collected, we do know now a lot about the biology of this canola flower midge. So we know that there's at least 2 generations per year. So looking at the emergence trap data that we collected, we have what we call spring cages here in the solid lines, and then summer cages. So spring cages were placed on soil that were previously canola the previous year. Summer cages were canola that same growing season. And what we can see is that there is a large peak probably towards the end of June in 2014 and a little bit later in July in 2015. But we can see that there's this secondary peak. And so this is the second generation of adults that are emerging. We also know a bit about its life history. So we know that the females lay eggs on the developing flower buds. So far, this is the only portion of the plant we have found eggs on. They have about 3 larval instars down here in the bottom left. They pupate within larval cocoons shown here on the right, and that's a pupae right here. So we know a lot about its biology. We also know that in Saskatchewan, we have at least 2 parasitoids that have been identified. So these small little black dots, they're not that recognizable, but these are parasitoids on these galled flowers. So if you were to open it, you would find larvae. And what we found is that there's 2 different species, an Inostemma species and a Gastroncistor species. And both of these species have never been found on Swede midge in Ontario. So this is interesting. This also might point to the fact that we have a different species of midge here on the prairies. We know the distribution. So this past summer in 2017, we conducted a very thorough kind of transect-based survey, east to west, north to south, across most of Alberta, Saskatchewan, and only in the Swan River Valley in Manitoba. So we need to really increase our sampling in coming years in Manitoba. But what we see is that damage with larvae, so those green points here, those are, we can positively associate those are caused by our canola flower midge. The ones in blue, those were we found potential damage. So it looked like the flower gull, but we didn't find any larvae. So it does appear to have a fairly widespread distribution across kind of that northern grain belt here in Saskatchewan and into east central Alberta. And so what about its damage? So as I've mentioned, the only damage symptom we've seen so far are these galled flowers or these bottle-shaped flowers. So these flowers, again, they don't open. Sometimes you'll see a pod growing out of it, but those pods usually have no seed in them. So it can be quite damaging if you have multiple flowers that are galled. So this one here on the right, you can see these dried older flowers down the raceme. This is kind of when the damage comes really obvious, is when you see these unopened closed flowers down the raceme, but you still see some flowers at the very top of the raceme. And if you were to open up these flowers later in the season, you'll see kind of this damage on the stigma here, these little black spots, and that's the plant trying to defend itself against these larvae. That's the typical damage it causes. So what do we know about its pest status? Well, unfortunately, I have to say it's currently unknown. We found this midge across a wide swath of Alberta and Saskatchewan, but the damage was very low. We were sampling about 100 racemes per field, and we were finding very, very few infested flowers. If you look at this picture, I apologize for the blurriness, but you can see these kind of darker, funky yellow, brown colors. Those are galled flowers. And this was kind of one of our worst fields that we saw. So we're continuing to conduct research, and we will hopefully figure out how much damage this midge is capable of causing. You do, see some scary looking things like this. If you get a raceme that, almost all the flowers were galled, well, clearly that could affect yield. But again, you're finding very few sites that look like this. And even within those sites, there's very few racemes that look like that. So again, we're not really sure of its pest status, but if we were getting really high populations and we were seeing multiple racemes like the one on the right, clearly that will have an impact on yield. We've also been proactive, and we're starting some pheromone monitoring. So we've identified the pheromone with the help of collaborators in the UK. We know that it's a 2-component pheromone blend. This differs in that swede midge is a three-component blend. And so this was just done last summer, but this summer, this 2018, we will be testing this in the field. And this will really help determine the distribution of this midge across the prairies, as well as any other region that we want to test. This will really help us find where it's located. All right. So just to wrap up a little bit, tell you what's happening in our future. Dr. Megan Van Koski, who's another research scientist here with Ag Canada in Saskatoon, and I have funded projects to continue to investigate both swede midge and this canola flower midge. So we hope to identify the distribution of both midge species on the prairies. So this will be really helped by the new pheromone that will be available soon. Well, we'll be testing out this summer. And we'll also continue to use swede midge pheromone traps and determine if we can find swede midge throughout the prairies. It's not that swede midge is not necessarily no longer here. It's just that the sites that we've been trapping at, it has not been present. So swede midge could still potentially be here on the prairies. We want to know a little more about its life cycle, so we're going to keep looking at its life cycle. We really want to see what the crucial stage is for when the female can lay eggs on the plant and what portion of the plant it lays its eggs on. We're going to look more into these biological control agents. Maybe these are really helping keep in check this as I mentioned, we've identified the pheromone, and so we're going to continue to use this for monitoring, and it's really going to help us determine the distribution of this midge across the prairies and maybe across the world. So with that, I just want to thank Dr. Julie Soroka, Lars Andreasen, Owen Olfert, and Megan Van Cossey for their help throughout this project. Jamie Heel and Dr. Rebecca Hall at the University of Guelph for helping us with the identification of this midge, as well as Brad Sinclair with the CFIA in Ottawa for the identification of this. Scott Mears and Shelley Barkley with Alberta Agriculture for conducting the Alberta survey for us. And then we have many wonderful technical assistants here in Saskatoon. And then funding for this was mainly provided by the Canola Council of Canada through their CARP program, which is co-funded by Canola, Alberta Canola Producers Commission, and the Manitoba Canola Growers Association. I also have one swede midge project that's funded by the Agricultural Development Fund and Western Grains Research Foundation, which also helped with this work. So with that, I'd be happy to take any questions.
Awesome. Thank you so much, Boyd. That was a fantastic presentation with lots of great information. I do have a couple of questions here. Apologies if you've already answered them. Some of them questions are from the beginning. You could have covered these throughout, but I think sometimes these questions are worth repeating. So someone had asked if the 20 midge threshold to spray is in the whole field, or is that per trap?
So that's based off of trap capture. So you'd have hopefully 4 traps in your field, and once the cumulative number of midge captured across those 4 traps is 20, then you would be looking to put on an insecticide application. Perfect.
And in the same realm as traps, so if producers are interested in monitoring for swede midge or for this new midge, where are they able to kind of start and get the traps and lures from specifically?
So there is a Canadian company, it's called Solida. So it's like the word solid with an A at the end. Solida.com. And if you look them up, you can get both the traps and lures from them. I will say that unfortunately, compared to other insect pheromone lures, the lures are a little more expensive for swede midge. They're about $13 a lure. And it requires, in order to monitor for the whole season, you're probably going to have to change those lures about 4 times. But those are commercially available, and if you think you might have swede midge issues, you should look into that. Also, if you're on the prairies and you're interested in monitoring for swede midge, send me an email, because we will be still conducting swede midge monitoring, and we can happily provide you with traps and lures to do some monitoring for us.
Perfect. So with regards to this undescribed canola flower midge, can Ontario maybe start to see that becoming a problem in the years ahead, or is that kind of out of your realm of knowing?
Yeah, right now we don't know. So as I mentioned, in Ontario with swede midge, the primary damage symptom they were seeing were the swollen and distorted tissue, and they very rarely see any type of galled or infested flowers. So swede midge is capable of infesting flowers. It's just very, very, very rare. And so, if it was present in Ontario, we would think that it's causing these galled flowers, but so far in Ontario, they're not really seeing it. So I guess that'll just be something to wait for the future and see if we ever see any galled flowers in Ontario, and then we'd have to determine what species is causing that, whether it is swede midge or whether it's this canola flower midge. Okay.
Someone had asked, in southern Alberta, we almost spray every year now for cabbage seed pod weevil at 10 to 30% flowering. Would this tend to control or minimize this new midge? He doesn't think he's seen as much damage here yet.
Yeah, so far we haven't really seen what we would call any positive damage associated with the canola flower midge in southern Alberta. With that insecticide spray, if there were these midge present, we'd really be looking to try and hit the egg stage or even midge forms, gull in the flower. They're really protected. So if there were eggs or adults present, the spray at that time could also reduce those populations.
Okay. And then someone from Saskatchewan specifically asks, we are seeing less plant damage here in Saskatchewan, does the spray recommendation still hold?
I would not be spraying for swede midge on the prairies. I don't think right now the damage, I don't think we can really positively confirm any damage we've seen on the prairies is associated with swede midge. And I don't think any of the damage we've seen associated with this canola flower midge would warrant spraying to date. And right now we wouldn't be able to tell you, at least with the canola flower midge, we wouldn't be able to tell you when to spray and if it would have any effect.
Mm-hmm. So someone asked if there's any work being done on temperature or moisture correlation to the hatching of pupae.
Yes, there's been some work done on that. So it's believed that there needs to be a significant rainfall event within 7 days in order for the pupae or the adults to emerge from the pupae. That, I should say, is with swede midge. We do not know what is needed for the canola flower midge, but we'll be looking into that hopefully.
Perfect. Just a comment here, I believe, to your question, Keith, about cabbage sea pod weevil. In Ontario, this is from another viewer here, he just mentioned that in Ontario, we often get a collateral control of swede midge when we are spraying for flea beetle here in Ontario. So I don't know if that kind of helps your question there, Keith. This looks to be one of our last questions to you, Boyd. Was there any damage that was checked as far as yield loss? And if there was it significant?
So not yet. So we have studies for this coming year and the following year after that to look at yield loss, potential yield loss. So that work will be ongoing.
Okay, perfect. Along with the rainfall event, is there a growing degree days that applies as well? Again,
I'll have to say not yet. We do have temperature data from both soil and air that we will be looking at trying to correlate with emergence of these midge.
So again, a lot of these questions will definitely be addressed once you guys do some more research here in the next couple of years.
Exactly. So we just wrapped up year one, and we have a couple more years of our current funded projects, and then we'll go from there and maybe have to.
Fantastic. And I'm sure our listeners can always count on Top Crop Manager to cover some of that research-based information. Well, thank you so much, Boyd, again for hosting our third session here in our webinar series. So I did just want to let everyone know that as far as our CAA credits go, you can submit credits to us by emailing your first and last name and your CCA number to webinars@annexweb.com, or by replying to the email that you will receive in your inbox with the recording, which again should appear within the next 24 hours. And the credits should be processed within the next 2 weeks or so. So again, thank you to our wonderful speaker, Boyd, and see you at our next session.
Thanks very much.